Good evening. I'm going to be giving a presentation on a case study that I reviewed for the YSTR and mitochondrial DNA in forensic analysis attached to this video. I'm also going to have the PowerPoint that I put together for my presentation. We are going to be having a case study in the realm of modern forensics, and within modern forensics, we're going to be talking about the analysis that provides accurate and dependable results for victims and suspects in forensic science, which helps hopefully lead to a conviction. The main focus of traditional DNA analysis is typically STR, or short tandem repeats. This provides each person a distinct genetic profile when looking at these small tandem or short tandem repeats. Other approaches, which STRs are not the only one, typically require, in situations where STR analysis is not practical or maybe not adequate, maybe the DNA was degraded, or there's a bunch of different reasons why STR would not be appropriate. This particular paper that I reviewed explores the functions of mitochondrial DNA and YSTR in forensic investigation and clarifies their use within the forensic realm and through this case study. DNA analysis has completely changed the field of forensic science by making it possible to accurately identify people. Now, this field is typically STR, short tandem repeat analysis, and it is most commonly employed in this approach. STRs are short chunks of DNA, which we previously thought to be trash DNA, and that repeat in particular locations throughout the genome leads us to this ability to be able to identify specific people. Even though STR analysis works really well, there are some scenarios within forensic science where it is difficult to produce a clear profile, which could be caused by deterioration or mixed materials. There's lots of different reasons why that would happen. Alternative techniques such as YSTR, maybe mitochondrial DNA can be used. They typically become invaluable when regular STR analysis fails to give us a result, or maybe it is not usable for the sample at hand. YSTRs are perfect for determining paternal lineage because they are found in the Y chromosome and are passed down unaltered from the father to the son. Contrarily, mitochondrial DNA is the opposite. It is transmitted exclusively from the mother and is found in numerous copies within the cell, which increases its resistance to deterioration, making it a better use for forensic results. These techniques are especially helpful when it's difficult to evaluate a mixture, when samples are damaged, or when unknowing paternal or maternal lineage is important for the case. If you have heard of the Idaho quadruple murder, the YSTR was something that was used to be able to link the father's DNA, which was collected from the trash, to the suspect, who we had a partial profile or an incomplete profile from a snap on the sheath from a knife. And there hasn't been a lot of details given. This is still an active case, so that's something I'm really interested in hearing about in the upcoming trial for that particular case. Now, moving forward, the Y chromosome short tandem repeats, or YSTRs, have special qualities that make them useful within forensic science. The Y chromosome's characteristics are passed unchanged from father to son, making it really helpful, especially when there are mixed information. This is something that I found interesting to read in the paper by Johnson and Smith. It's called YSTR Analysis Solves a 20-Year-Old Sexual Assault Case. It was a really interesting read and led to some really beneficial information on how that case was solved, which I'll discuss here in just a little bit. The Y chromosomes or the YSTR are an efficient way to trace paternal lineage because they are passed down and they are not altered. This is particularly helpful in situations when there's maybe male and female DNA that's mixed, maybe in a case like a sex assault where we did a rape kit and got both contributors. This is really helpful to isolate the male DNA, and then we're able to then trace it through YSTR. The capacity to trace paternal lineage using YSTR helps reduce the number of potential suspects and then helps us to narrow in our investigation. Now, contrarily, if we're looking at mitochondrial DNA, like I basically described previously, only the mother can give mitochondrial DNA, and that's passed down through the matriarchal line, which is located in the mitochondrial DNA outside of the cell's nucleus. So, of course, if we remember basic biology, we have a cell. The nucleus is typically considered in the middle. It's surrounded by a nuclear envelope, which is kind of like a cell membrane within the cell, surrounds the nucleus. So outside of that secure nucleus area, we have a mitochondria. The mitochondria does have some protection on it, but it carries its own set of mitochondrial DNA. Now, that mitochondrial DNA has several copies within the cell within that mitochondria, and it is more abundant and simpler to extract and is typically less degraded. Because of this property, being more plentiful, being less degraded, being a little protected, the mitochondrial DNA can be really helpful in cases where we need to identify maternal relation. Things like cases of missing children or missing persons, where a standard DNA, nuclear DNA addition or sample would be tainted maybe. The mtDNA or the mitochondrial DNA would be a great place to look for that information. In cases where there's maybe alternative DNA sources that are not feasible, maybe remains, those can also use mitochondrial DNA. So talking about that case study that I mentioned earlier in the Johnson, Smith, and Lee paper, basically the YSTR analysis solves this 20-year-old case in which Johnson, Smith, and Lee published to the Journal of Forensic Science in 2023. And they demonstrated the ability to use YSTR analysis. In this instance, a sexual assault that happened in June of 1998 happened in a Colorado town, and the female victim was left unidentified for 20 years. I can't imagine having a family member that was left unidentified for that long and just not knowing. The offender was never found, and in 2018, the case was revived or revisited. It had gone cold, and from my personal experience with cold cases, they typically sit in a closet and a new cold case detective comes on, and then those kinds of things are revitalized, or they start looking into cases that have sat stagnant for a long period of time. And that's what happened in this case. In 2018, the case was revived because of improvements to forensic DNA. They were able to take the DNA samples that they had from the scene or from the rape kit and several other contributors, and basically they were able to do a YSTR analysis, and they were able to take that DNA sample. And in 1998, the assault case had a combination of a victim and the attacker DNA, and that made it a little difficult. This combo in 1998 was not easily separated as it is today. Traditional STR analysis was greatly hampered or slowed by the sample's low concentration of male DNA, which made this even more difficult. There wasn't enough DNA to get a full STR profile, so they did get a partial, but it wasn't enough. It was too intermingled with the other DNA from the female. These restrictions made the inquiry more difficult and prolonged the time that it took for them to find a suspect profile. The YSTR analysis that was used to re-examine this evidence was basically involved amplifying the STR markers that were specific to the Y chromosome, which we've looked at in class, and it extracted DNA from the preserved material. Then it took a YSTR database and matched and amplified these markers. By using this specialized technique only for YSTR, the forensic experts were able to get a more robust profile. They were able to separate the male and the female from the mixed sample, and basically they were able to obtain a distinct genetic profile that was then linked to the father, because of course YSTR is passed down through the father's line. Then they were able to take that sample of the perpetrator's paternal lineage and isolate the male profile, and they were able to get a match. And in the match, they were then able to make a suspect identification through this analysis. They were able to corroborate the evidence, and the obtained evidence from the scene and all of that additional investigation and this new technique. An arrest was actually made 20 years later. A 20-year-old cold case was then resolved, basically highlighting the value of re-examining historical evidence, as long as that evidence was properly preserved. And that's a whole nother issue and a whole nother presentation. So basically, in forensic science, the role of YSTR and mitochondrial DNA tests are essentially indispensable. Basically, these are instruments that we're able to enhance conventional STR analysis. We're able to take that initial amount of STR data that we get, and we're able to make it, through these techniques, adequate enough for us to start making links and solving cases. These methods have really helped to provide a whole new set of possibilities for identification, especially within cold cases. A lot of cold case detectives are really starting to rely on YSTR and mitochondrial DNA analysis. I think that this is going to make a huge breakthrough. I think we'll start to see a lot more cold cases solved through the use of YSTR and mitochondrial DNA. And this article by Lee, Smith, and Johnson was really interesting in that it really detailed the scientific aspect. So I highly recommend taking a look at that. Thank you so much for listening to this presentation, and have a wonderful day.